This is a model of DNA, the first component of the strands. This DNA molecule consists of one nitrogenous base, one phosphate group, and deoxyribose sugar. These three things will make up a monomer of DNA called a nucleotide. The polymer is DNA. DNA replication occurs during the cell cycle process known as synthesis. During synthesis, DNA is identically replicated, so when the cells split, it has the same amount of DNA in each cell. This phase takes up half of the time in the cell cycle because of how complicated it is. During DNA replication, the DNA helicase splits the parent strand of DNA down the middle, creating two half strands. This allows polymerase to add nucleotides to each side. Once DNA splits, there are two strands, the leading strand and the lagging strand. Both of these strands move from 5' to 3'. The leading strand moves with the rest of the DNA. As it splits, the lagging strand moves towards the end of the DNA, causing it to have to be split up into Okazaki fragments. This makes these strands anti-parallel, which means the strands are parallel but facing opposite directions. After the DNA strands are separated, primase proteins begin traversing through the strands. It distributes RNase primers, an anchoring place for the new nucleotides. On the leading strand, it only has to lay a little, but in the lagging strand, it lays a lot. These segments each have the sum primer because it needs somewhere to attach. After primers, DNA polymerase go and start at the primers. With placing, it is grabbing free nucleotides and leading it into match adenine with thymine and cytosine with guanine. Then, if there are any gaps, they are filled with ligase, which fills any gaps. This process eventually creates two DNA strands, each with a parent and daughter strand. This is called semi-conservative. This is how DNA replicates.